Hello friends, I am Manju Sharma. Today we will study about development of embryo or embryogeny in dicot plants. So first of all, what is an embryo? The embryo is the part of seed or bud that contains the earliest form of plant's root, stem and leaves. The embryo develops at the micropylar end of the embryo sac where the zygote is situated. Now, what is embryogeny? The embryogeny refers to the development of the mature embryo from the zygote. So, in this video, we will study about the embryogeny in dicot plants. The first division of the zygote is almost always asymmetrical and transverse to its axis, producing a small apical cell and large basal cell. This division provides polarity to the embryo. The apical cell divides vertically and the basal cell divides transversely to form four-celled structure. This structure is called as proembryo or it is T-shaped. The upper three cells of proembryo divide further by anticlinal and periclinal division to form 32-celled globular proembryo. The outer layer of the globular proembryo develops into a distinct layer called dermatogen, which later forms epidermis, and the inner cell of the upper region develop into the cotyledons and plumule, while the lower cells develop into the radicle. The cells of cotyledons divides rapidly than the plumule cells, and therefore the embryo become heart-shaped. When the cotyledons are completely formed, they enclose the plumule at the base, and the plumule is also known as shoot meristem. The basal cell undergo a series of transverse division resulting in the formation of hypophysis and suspensor. Now, what is hypophysis? The hypophysis is the uppermost derivative of basal cell, and these cells serve as a precursor to the root cortex initials and central region of root cap. Now, what is suspensor? These are an empirical embryonal structure and comprises of single row of 6 to 12 cells. Now, what is the structure of dicot embryo? The two embryonic leaves called cotyledons attached to the embryonal axis. the embryonic shoot called plumule, and this develop into the shoot, and the embryonic root apex called radicle is developed into the root in future, and the part of the axis above the radicle is called hypocotyle, and below the plumule is called epicotyle. Now, what are the anticlinal and periclinal division? The anticlinal division are perpendicular to the adjacent layer of the cells. So, what we get is columns of cells adjacent to one another. And the periclinal divisions are those divisions that occur parallel to the tissue or organ surface. As a result, we get row of cells stacked one over other. In the next video, we will study about embryogeny in monocotyledons. Till then, bye bye and thanks for watching my video.